Welcome to the BioInsights, where science comes alive.
50%. I want you to really just sit with that number for a second. If we look at the 10 most commonly prescribed medications in the US right now, at least half of them, literally 50%, do the exact same biological trick. They are all enzyme inhibitors. Which means getting a grip on this one single biochemical concept, that's the key to understanding how we treat everything from high cholesterol and bacterial infections, all the way to severe pain and high blood pressure.
Today, we're diving into something I like to call the microbattlefield. We are going to explore the mechanisms of enzyme inhibition, which is basically the story of how humanity figured out how to hijack the biological brakes inside our cells to actually cure diseases. It's a wildly fascinating look at the microscopic machinery keeping us alive, and exactly how we strategically shut it down when things go off the rails.
Okay, let's jump right into the agenda so you know exactly where we're headed. First up, the brakes on biology. Then, we'll talk about suicide inhibitors. Third is fighting for space. Fourth, changing the rules. And we'll wrap it all up with the drugstore connection.
All right, part one, the brakes on biology. Now, before we can really grasp how these drugs work, we kind of need to understand how biological speed control works in the first place, right? Our cells are absolutely jammed with these tiny protein machines called enzymes, and their whole job is to speed up chemical reactions. But, well, sometimes the reaction is happening way too fast, or just at totally the wrong time. And that brings us right to our core definition. An enzyme inhibitor is literally any substance that can decrease the velocity of an enzyme-catalyzed reaction. So if enzymes are the accelerator pedal, inhibitors are the brakes. They physically interact with the enzyme to slow it down or just stop it completely. And biology actually has two very different ways of slamming on those brakes.
Over on the left, we've got irreversible inhibitors. These guys form permanent, incredibly strong covalent bonds with the enzyme. It is honestly like pouring quick-drying concrete right onto a brake pedal. For instance, this is exactly how lead poisoning works. Lead forms these permanent bonds with protein side chains, completely destroying the activity of an enzyme called ferrochelatase, which you absolutely need to make heme for your blood. But then, on the right side, we have reversible inhibitors. These form temporary, non-covalent bonds. So if you just dilute the environment, these inhibitors literally wash away, and the enzyme springs right back into action like nothing ever happened. Moving on to part two, the suicide inhibitors. So within that pretty terrifying category of irreversible inhibitors, there's a specific subgroup with, honestly, one of the most hardcore names in all of biochemistry. And what's wild here is the sheer level of deception involved. Suicide inhibitors, which scientists also call mechanism-based inhibitors, they don't just attack the enzyme head-on. No, they actually disguise themselves as normal substrates. The enzyme picks up the inhibitor, totally fooled, and actually starts to process it. But in doing so, the enzyme converts that inhibitor into a highly reactive form that instantly and permanently locks right back onto the enzyme itself. The enzyme literally triggers its own destruction. It is such a cinematic, almost sci-fi way of shutting down a biological pathway.
Let's look at part three, fighting for space.
Let's take a step back from that permanent destruction and look at temporary control. This is called competitive inhibition. And the concept here is wonderfully simple. The inhibitor and the normal substrate are basically fighting for the exact same parking spot on the enzyme, which we call the active site. If the inhibitor gets there first, it physically blocks the substrate from binding. Now, one brilliant way scientists exploit this is by creating things called transition state analogs. These are super stable molecules perfectly designed to mimic the exact structural shape the substrate takes right in the middle of a reaction. Because they look so much like that perfect intermediate stage, they bind to the enzyme's active site with a much higher affinity than the normal substrate ever could. They basically win that fight for space every single time.
So the really important thing to grasp is how this literal fight for space changes the enzyme's kinetic math, its speed and efficiency. With competitive inhibition, the Vmax, which is the absolute top speed those enzymes can reach, stays completely unchanged. Why? Well, because you can actually overwhelm the inhibitor just by flooding the system with normal substrate. If you've got 1 million normal molecules for every one inhibitor, the normal molecules are going to win the parking spots and the enzymes will run at top speed. However, the apparent Km is increased. Km is basically a measure of how much substrate fuel you need to get those enzymes running at half their top speed. Because that inhibitor's in there competing for space, you suddenly need way more fuel just to outnumber the competition.
All right, part four, changing the rules. Now, what happens if the inhibitor just completely refuses to fight for that active site parking spot? What if it uses a totally different tactic?
Well, that's non-competitive inhibition. In this scenario, the inhibitor binds to an entirely different site on the enzyme. It's essentially a flank attack. And because it's not fighting for the active site, this non-competitive inhibitor can bind to a totally free enzyme, or it can bind to an enzyme that's already perfectly locked onto a substrate. It honestly doesn't matter. The second this inhibitor attaches to that side spot, it alters the enzyme's overall structure just enough to completely shut down the chemical reaction.
Because the rules of engagement here are totally different, the kinetic math just flips on its head. With non-competitive inhibition, the apparent Vmax, that top speed we talked about, is permanently decreased. And you can't just overcome this by adding more substrate because, remember, the substrate and the inhibitor aren't fighting for the same spot anymore. Flooding the system does absolutely nothing to knock that inhibitor off its separate site. Those working enzymes are basically just taken offline. But, and this is the really fascinating part, the apparent Km is completely unchanged. The inhibitor doesn't mess with the substrate's ability to bind to the active sites of the enzymes that are still actually working. So the fuel efficiency of those remaining uninhibited machines, it stays exactly the same.
And this brilliantly illustrates exactly how scientists figure out what kind of inhibitor they're dealing with in the lab. They use these graphs called Lineweaver-Burk plots. You can really just think of these as chemical fingerprints. If you look at the left side for competitive inhibition, because the top speed, or Vmax, is unchanged, the lines for the inhibited and uninhibited reactions perfectly cross each other right on the y-axis. Now jump over to the right side for non-competitive inhibition. Because the top speed drops, but the binding efficiency, the Km, stays the same, those lines intersect at a totally different spot over on the x-axis. Literally just by glancing at where these lines cross, a biochemist instantly knows the tactical rules of the microscopic battle they're looking at.
Finally, Part 5, the drugstore connection.
Okay, time for the grand payoff. We're going to take all this abstract kinetic math and these biochemical rules and see how they directly save lives every single day. Starting right at the top row, statins, like pravastatin. These are competitive inhibitors. They're structural analogs that directly compete for the active site of HMG-CoA reductase, which is basically the bottleneck enzyme in making cholesterol. By winning that fight for space, they lower your blood cholesterol. Next up, penicillin, a classic antibiotic. This works by completely inhibiting the enzymes that bacteria use to build their cell walls. Then you've got blood pressure meds like ACE inhibitors. Lisinopril is a great example. These block extracellular enzymes. Specifically, they stop the angiotensin-converting enzyme from making a potent vasoconstrictor. You block the enzyme, dilate the vessels, and boom, lower blood pressure. And think about just everyday aspirin. It irreversibly inhibits the synthesis of prostaglandins to stop pain and inflammation. It's basically pouring concrete right on the pain pathway's brake pedal. And wrapping up the table, oxypurinol, a medication for gout, acts as a non-competitive inhibitor on an enzyme called xanthine oxidase. It binds to the side, completely changing the rules to stop purine degradation. So all of these radically different diseases are being treated by the exact same microscopic mechanisms we just unpacked, which leaves us with a pretty incredible thought to close on. If we've already figured out how to master these biological breaks to invent things like statins, penicillin, and aspirin, literally the bedrock of modern medicine, what cellular machinery are we going to learn to perfectly control tomorrow? The possibilities are just endless. Keep exploring, keep asking questions, and seriously, never stop marveling at the machinery inside of you.